Today we're going to start talking about more about enzymes and how they pertain to our discussion of thermodynamics that we did on Monday. Now, we have talked about enzymes for a while. We've talked about different reactions that enzymes perform, and we have talked about the connection between structure and function. And what you're seeing here are two different theories that are slightly different, but they have really important differences on the form and the function and the shape of enzymes. So take a close look at these two. And if someone will write in the chat box, what's different about this model up here? The enzyme is the E, the substrate is the S, and the products are the P's. What's the difference between this and this one here? Okay, so we have a suggestion that the enzyme at the transition stage is different between these two. And that's a really good observation. So the enzyme at the transition stage is different. So because as we see here, the enzyme becomes rounded, whereas here in this other depiction, the enzyme does not change shape at all. And this is really important because the enzyme changing was originally not predicted in the bottom lock and key model. So this one was earlier. This was the earlier version of the theory, and this is the more modern. Sorry, it's a little sloppy, but again, the top one is more modern, the bottom one is earlier, it's less modern. And originally, scientists did not know that enzymes could change shape. They didn't know that enzymes had different conformations. And we've talked a lot about different conformations of enzymes, of proteins. And so scientists originally did not know anything about those different conformations. The reason why was that they weren't looking at enzymes in different situations. They were crystallizing enzymes and then analyzing those shapes. So it makes sense that they only predicted that there was a single shape for an enzyme. That was the enzyme when it's unbound, when it's bound, and then after the products have been made. On the other hand, now we know that the induced fit model at the top is more accurate. So the induced fit model shows that the enzyme changes shape, and also, interestingly, the active sites or the binding sites of the enzyme are different in these two models. So we see that the active site in the lock and key model fits the substrate exactly, right? Like it has this little, almost like a rounded arrow shape. And it fits the substrate exactly. And that might be what you would expect to see. Like, of course, the enzyme is going to be complementary to the substrate, right? But in reality, it's not exactly complementary to the substrate. What we see is that definitely has some complementary features to the substrate, but it doesn't match the substrate exactly. It actually matches the substrate transition state a lot better than the substrate itself. And that's because the enzyme will change shape a little bit, changes conformation as the substrate changes conformation. So it's kind of like a cooperative process in the induced fit model. And induced means something that is kind of encouraged or persuaded into doing a certain thing. And so you could kind of think of the enzyme changing shape slightly because it's encouraged by the substrate. The other thing I want to point out, which is true of both models, is that the enzyme returns to its original state. So one of the key characteristics of enzymes is that they can be used over and over again. And enzymes, therefore, need to return to their original state after they perform a reaction. They're not going to have lost any atoms or any hydrogens themselves. They're not going to have broken any bonds or formed any bonds within the enzyme that cannot be reversed. Okay? And this, if you're interested in disease science or pharmaceuticals, this is a really important principle. If you recall, when we talked about antibiotics, we said that antibiotics, or some antibiotics at least, target the ribosome, and they'll just stay bound to the ribosome. And that prevents the ribosome going back to the original state. And so that is actually how a lot of pharmaceutical drugs work. They will prevent the enzyme from returning to the state where it's able to bind to new substrates. And it's pretty simple, you don't need to break an enzyme, you don't need to take it apart. All you need to do is make it stay bound to something to stop it from working. Oh, oops. So this is just a depiction of how much an enzyme can help. So we've talked in the past about some different types of reactions, and most of these reactions you can imagine would not occur without an enzyme. You're not going to get the polymerization of DNA without an enzyme. You're not going to get the formation of RNA or the reading of RNA and creation of a protein without an enzyme. But you might get a single reaction. So for example, without DNA polymerase, maybe we would get, two single nucleotides bound to one another. And that's a reasonable assumption to make. That might happen without the help of an enzyme. However, that reaction going forward would be very, very slow. So this this depiction at the top here is the amount of products that would be formed without an enzyme. So for example, if we were talking about the synthesis of DNA, without DNA polymerase, we might get two nucleotides linked. Out of all the nucleotides total that possibly could be linked, which would occupy this entire box, this many nucleotides would end up being linked after 1 year. So it's not very many, but it is some. However, if we look at the bottom, then all we need is 1 second with 1 molecule of DNA polymerase to link all of these nucleotides. Enzymes work very quickly, and they're very efficient at making their reaction go forward. And so that was just, a kind of cartoonish depiction, but we have actual data on the rate of reactions with and without enzymes. So these are some reactions that would occur spontaneously. And we see in the green bars the enzymes that would normally synthesize that reaction or make that reaction go forward. So these bars on the right side show the actual time that it would take the enzyme to catalyze that reaction, one reaction. And if we compare that to the bars on the left, those are the amount of time that it would take for that reaction to occur spontaneously. So let's look at this one specifically, adenosine deaminase. So this is an enzyme that modifies the adenosine nucleotide. And this enzyme can perform that reaction in a little bit over 1 millisecond. So you get many of these reactions in 1 second. On the other hand, if we didn't have this enzyme and we wanted that reaction to go forward, we would have to wait well over a year, somewhere probably around maybe a couple hundred years for that reaction to go forward randomly. So reactions can occur randomly, and this is related to the study of the beginning of life on Earth, or possibly life on other planets. You can have purely abiotic reactions occurring, but they do not occur often. So if you find a lot of molecules that could only result from a certain reaction, it probably means there's an enzyme present. So as we saw in that depiction of the induced fit model of enzyme-substrate relationships, enzymes bind substrates transiently. And that just means that they bind enzymes and then they release them. So there's a brief encounter where we have the enzyme and the substrate bound, and then we end up having the enzyme and the product. At least that's what we want in the natural or the native reaction. And this is a slide I've used in a biochemistry class, so you don't need to pay attention or memorize all this stuff, particularly like all the technical wording here. But hopefully, if you read it, it might give you a little bit more insight. But I do want us all to pay attention to this graph. And we looked at a form of this graph on Monday as well. So in this graph, we have the amount of enzyme along the x-axis, so the amount of enzyme. And I don't know, some of you have probably taken chemistry or are taking it when things are in brackets. That is the concentration of whatever enzyme or product or reactant you have in your mixture. And on the y-axis here, we have velocity or the rate of reaction. And we see that this graph is illustrating basically a situation where we are adding more and more enzyme to our reaction mixture. So we start out with no enzyme, and then we increase the amount of enzyme across this y-axis, and then the reaction rate increases. So we're not measuring product exactly, we're measuring the amount of product produced per second or per millisecond or whatever. So the more enzyme you add in, the faster that reaction is going to occur, the more reactions you're going to have per second. And that makes sense if we take another look at this other graph here. As you add more enzyme, or if you compare enzyme versus no enzyme, then we see that, of course, the reaction rate increases exponentially, but also if you add more enzyme molecules, then you're going to increase the reaction rate. So remember, in this example here, we're only adding one molecule of enzyme, I believe. Or no, we're looking at the reaction rate. So that reaction rate is this for no enzymes present, and then with enzymes present, we would have this reaction rate. And so it's related to this graph, again, where we add more and more enzyme, and then we get a faster and faster rate. And the rate is very, very low, as we saw on the previous slide, if we have no enzyme at all. I also wanted to touch on this idea on the next slide. So this slide shows the differences in enzyme function at different temperatures and different pHs. So let's take a look at the temperature first. So again, we're looking at the rate of reaction, and that is on the y-axis, instead of the concentration of enzyme on the x-axis, we have the temperature. And that would be the temperature of the reaction mixture. So for a typical human enzyme, we have the highest rate at around 37 degrees Celsius. Does anybody want to guess why that is? Anybody? Why do we think that we have the peak activity at this temperature? Yep, exactly. Yes, Melissa and Steve, you're correct. So this is the typical body temperature for humans. And so your enzymes work best when you're at the correct, so to speak, body temperature. And this is one of the reasons why sometimes things will not work correctly if you have the wrong body temperature. So if, for example, if you're very cold, hypothermia, not everything is going to work correctly in your body. If you were extremely, extremely hot, things would not work correctly either. And in addition, there are slightly different temperatures for some different parts of your body, and some enzymes that are in a specific part of the body will also work best at that temperature. But this is just in general for the average enzyme in your body. Now here we see a different graph, and it says that this is the optimal temperature for a typical enzyme of thermophilic heat-tolerant bacteria. And so this is obviously much higher. It's a little bit below 80. And where have we seen the use of one of these thermotolerant or thermophilic heat-loving bacteria? Well, we've seen these in our discussion of PCR. So in PCR, we talked about Taq polymerase, and the Taq stands for Thermophilus aquaticus, and that was hot spring bacteria, and we isolated its DNA polymerase, and we used that for PCR, and we could use a human enzyme, but the really nice thing about using the bacterial thermophilic enzyme for PCR is that we could keep it from working until we know that the primers are annealed, we already have nucleotides in our solution, et cetera, so it's not going to start working until we want it to. So it's not going to start making a bunch of DNA that we don't want. So now let's go to the bottom here, where we see a similar graph, a similar illustration, but this one, instead of being for temperature, is for pH. And we have a similar phenomenon that occurs in enzymes with respect to pH. Enzymes are often only folded correctly if they're at a specific pH. And we're not going to go too much into this, but it has to do with, of course, protonation and deprotonation and the different groups on the amino acids. So for most enzymes, we would expect there to be a normal pH or a neutral pH. So this might be for an average enzyme in your body, like DNA polymerase maybe. But then we see on the left and the right, we see these two specific enzymes, pepsin and trypsin, which are located in specific parts of the human body. And in those specific parts of the human body, those organs have unique pHs. So the organs are kept at those pH. And so the enzymes that have a function there need to be able to perform their reactions at that pH. And if you put them at a different pH, they would not have a high rate of reaction at all. They might become unfolded and might not be able to function. So we need to keep all of these things in mind when we talk about enzyme function. And as we move on, we're going to be talking not only about the function of one enzyme, but the function of many enzymes as a group. So, got a bit of a mess here. So actually, I might want to come back to this slide in a couple minutes, but what I want to point out here is, again, the use of ATP is very important in terms of using energy in the cell. And as we've discussed, the chemical group on ATP which leaves is the phosphate group. Sorry, I'm going to move this to this screen so it looks like I'm looking at you guys instead of ignoring you. Not ignoring you guys. Okay, so Now we're going to start talking about a little bit more advanced stuff. We're going to talk about how our complex reactions that we need to be performed are performed even though they might not be favorable reactions. So how does that occur? We've been talking about different values for the change in free energy, or the delta G. So a positive delta G is that a favorable reaction or unfavorable? Positive delta G, being positive is actually bad. Being positive makes me negative, makes me, I hate being positive, because that means that it takes energy. And that, actually, you can kind of relate that to your regular life, especially right now. It takes a lot of energy to be positive right now, doesn't it? It's just the year 2020, it takes a lot of energy to be positive. And so you need to put energy into that positivity. And so the same thing goes for reactions that have a positive value for delta G. It means that we need to put energy in. They use energy. And overall, at the end, after the reaction has gone forward, you have used energy. You have less free energy than you did before. And I believe that if you guys were interested, I don't have it here, but there was a slide, and I could repost it again later, that has a link to a video that goes through why it seems a little bit weird, and it seems like the positive should be negative and vice versa. It's just the way the formula is derived. And again, I'll post that link if you're interested in the math. That might help some people. I'm not a math person, so didn't really help me, but I'm happy to provide it for people who might be interested. Okay, so this reaction in particular is the production or the synthesis of sucrose. So sucrose is a sugar. It is just a glucose molecule bound to a fructose molecule. So these all might seem familiar. They are carbohydrates that we have in our food. And then my analogy, that has a positive delta G, that my analogy for that is making a car move. And without the enzyme to make the car move, you'd have to be pushing it on your own, and that is pretty tough. That might be a positive change in Gibbs free energy. You need to put a lot of energy into it. However, we have some tricks up our sleeve. We have ATP. ATP hydrolysis, as we've talked about many times, has a negative delta G. So ATP hydrolysis is a favorable reaction. So it does not require energy, and we phrase it as it releases energy. What it really means is that we have a favorable reaction where the products are more stable than the reactants. Okay. And then our comparison here is the burning of gasoline. And of course, that is a favorable reaction. Again, not necessarily spontaneous. Your gasoline is not going to burst into flames by itself, hopefully, unless you're in like a James Bond movie, crazy things happen. But once you put that gasoline in the car and the reaction occurs to start burning it, combusting it, then you have a favorable reaction that has a negative delta G. So what happens in the cell is that unfavorable reactions are normally kind of broken up into a series of steps. And sometimes each step is favorable because of the way that it's broken down. Or sometimes some of the steps are favorable, some of them are unfavorable, so we need to use ATP. But overall, if you look at the net, then you're going to have a negative delta G. That is the release of free energy. Does not require energy. If you do it in this specific sequence. So what we see here is glucose. So again, our goal is to make sucrose. But instead of starting just with glucose and fructose and sticking them together, which is an unfavorable reaction, what we have is glucose and then the use of ATP. So we hydrolyze ATP, and what happens is, we've been talking about hydrolysis of ATP, and normally we drive some reaction forward, and it's just kind of like fuzzy, like how does it do that? I don't know, we don't care, it's magic, right? Actually, not magic, this is actually a science class, you guys didn't know. What happens is that we hydrolyze ATP, we break off that phosphate group, and add it onto glucose. So once we add it onto glucose, we know that phosphate groups are good at leaving because they can kind of balance their own charges. And so now this molecule, this product, glucose 1P or glucose 1-phosphate, has this good leaving group. So this reaction has a negative delta G because we've used ATP, right? And we've produced ADP, and then that other phosphate group goes onto glucose instead of like floating away. Now we can take glucose 1P and remove that phosphate group, which is also a favorable reaction, and use that energy to form a bond between glucose and fructose to form sucrose, and then we release the phosphate group. So that also has a negative delta G. It is a favorable reaction. And now we've made sucrose, and overall we have a negative delta G value. So it really matters what order you do the reactions in. And again, oftentimes for these types of things, you need to use some ATP. And the other thing that I point out here is that the products become the reactants for the next reaction. And we're going to see this, if you guys remember, with photosynthesis and cellular respiration, you have all these steps, and it's like taking, for cellular respiration at least, it's like taking a glucose molecule, and it goes down an assembly line, basically. You do a little bit to it in this reaction, the next reaction it tweaks it a little bit more, and so on. So again, the products become the reactants for the next reaction. So I love this slide. If you don't have a high quality connection, you might not be able to see it well. So you might want to go and look at the slides afterwards just to see this, just to look it over. This slide shows the metabolic pathways just for human lung inflammation. So not even normal lung function. These are the enzymes that are active during lung inflammation in humans. All of the dots are going to be products, and the lines in between them represent reaction of their enzymes. And these are color-coded, so each different type of pathway, set of reactions, is a different color. So for example, in pink we have energy metabolism. In orange, we have amino acid metabolism, so using amino acids or making amino acids. Carbohydrate metabolism is shown in this blue, so that would be use of sugars or construction of sugars if we needed to construct sugars in order to make, like an amino acid or something. We see some things that we might recognize here. So you may have heard of the citric acid cycle, the TCA cycle. We're going to talk about that in detail in probably next week's classes. That is something that occurs in the cell. And also carbon fixation you've probably heard of, so another common set of reactions. my point in showing this to you is that there are so many reactions going on in the cell at once, and the products and the reactants for different types of reactions even are connected. And sometimes there are a lot of different options as to where a product can go next. So for example, in the citric acid cycle, which is purple, we have this nice circle here, right? But also we have connections to this dot here. So the products could go into this other set of reactions where we're using alanine and we're using lysine, or it connects to this gray line here, which is pyruvate metabolism. So there are lots of different connections, and different reactions in the cell might affect one another. So this is a really complex area of research. It's really interesting. If you're interested in this type of thing, sometimes this is referred to as network analysis. This would be a network of different enzymes. And so they actually, I was reading a news release yesterday, I think, 2 days ago, that used network analysis to try and figure out which reactions in the body that are optional, in other words, which reactions that could be affected either by you taking medicine or by you eating something or being hot or cold or tired or whatever are related to resistance to coronavirus. So these types of schematics can be really powerful for generating ideas. You do need to test them in the lab because, as you can see, with so many options, you can get things that just randomly come up as looking significant when they're not. I won't get too much more into that, so I know that get a little bit overwhelming. And what forms? I didn't say that. Don't tell my former boss I said that. Okay, so what we've been talking about basically in the past couple slides is the linking of reactions that have a negative delta G, or are favorable reactions, with reactions that have a positive delta G, or are unfavorable. And so it's similar to when you use a water wheel, or when they use the water wheels to, they use the flow of the water and they harness that to power other reactions like the turning of a millstone. And so the cell also does this and uses favorable reactions to fuel unfavorable reactions. Okay, so we've already talked about this. So we're going to be talking about photosynthesis. And photosynthesis is obviously very important for plants, but overall for life on Earth. Not to exaggerate at all, but it is. The general summary of photosynthesis is that cells will absorb energy, or photons of light, and they use that energy to build ATP. We'll talk about the different reactions that the cells need to perform in order for this to happen. Then we also have the use of ATP to make glucose. So we've talked a little bit about both of these types of molecules. ATP is a really commonly used molecule for actually being used by enzymes. Then on the other hand, glucose is, we've never seen a glucose molecule bound by an enzyme and used to fuel a reaction. Like we've been saying, DNA polymerase, it binds to ATP or GTP or CTP or TTP, but it doesn't bind glucose and use that to fuel a reaction. But glucose is a really good storage molecule for energy. So instead of having a ton of ATP in the cell, where any enzyme might be able to use it, even if they maybe shouldn't at that time, our cells store energy as glucose, and plant cells do as well. And I'm going to mainly talk about plant cells when we talk about photosynthesis, but I do want you to know that other types of organisms also can perform photosynthesis. So there are many bacteria that are photosynthetic, and there are some other symbiotic organisms, like some sea slugs, that are also photosynthetic. And then there are even more organisms that can fix carbon, that is, which can make glucose from inorganic carbon in the environment, or specifically carbon dioxide. And then the opposite of this is cellular respiration. So this is the breaking down of glucose, and all organisms do this. All organisms can break down glucose to make ATP, which can then be used by individual enzymes in the cell. And then the byproducts normally of cellular respiration are carbon dioxide and water, as well as ATP. And so we have all the different organisms here, lots of different organisms. Okay. So we haven't talked too much about plant cells. We've talked about them a little bit in the past. We know that they have a cell wall, so they're rigid. They look different than animal cells. So most of you were able to look at plant cells under the microscope in our last lab. We saw a lot of repeating structure overall in the cells because we have those cell walls set up, and then the cell membranes are within those. And so we have a lot of structure to plant cells and plant tissues overall. And I don't think that we were able to see the chloroplasts necessarily, because in that last lab we were taking an imprint of the cell. So we weren't actually seeing the color of the things in the cell. But if you were able to look at a plant cell, a fresh one, then you would be able to see the chloroplast. And that's what we're really interested in, because that is where photosynthesis takes place. And then another important feature of plant cells is this vacuole. And a vacuole is where water is held in a plant cell. And if you don't water your plant, like I sometimes do not do, then all the water is going to be used up in the vacuole, and then your plant starts to wilt. And that's also why you would kind of get your plant to perk up very quickly by watering it, because if you water it, then it just sends water through and fills up those vacuoles, and it will make the cells kind of blow up again. Okay, so in this view, I'm just blowing up this region right here. So our chloroplasts are shown here, and we see here that this is a zoomed in version. So like the mitochondria, as we'll see next week, the chloroplasts are these organelles that have a membrane that surrounds the entire thing, but then they also have a membrane inside. So this white part is the outer membrane. And then there's the inner membrane, and inside of that is the chlorophyll-containing thylakoid membranes. So what we're seeing here is really just the double membrane around the outside, and then interior to that are these chlorophyll-containing membranes. So in this image, it's not actually correct because we see the dark green outline of these chloroplasts, but really that shouldn't be dark green. The green color comes from the inner membranes. So again, the inner membranes are the ones that contain the chlorophyll. And so that's where this whole process is going to start, where we are going to see the absorption of that light energy. And so this is a depiction of a plant cell, and we see that there are chloroplasts within the plant cell. So that's another important point. A plant cell doesn't have just one chloroplast. In this depiction, it only has two. but in reality, a plant cell can have many, many chloroplasts. And then within each chloroplast, you have lots of the chlorophyll-containing membranes. All right, now let's talk about being green a little bit. So our human eyes see plants as being green normally. That's because the chlorophyll is green. Why is chlorophyll green? It's because it absorbs other wavelengths of light and it reflects green light. Now, why does it do that? That is because of the particular shape of the chlorophyll molecule. It allows it to absorb certain wavelengths, but not other wavelengths of light. So again, it reflects whatever color light we see. So if I'm looking at a green leaf, it's reflecting green light. If I'm looking at my gray cat, then it's reflecting wavelengths, many wavelengths of light to mix together and make that gray. If I am, looking at an egg, then it's reflecting back yellow wavelengths of light at me, particularly the U. So it's never absorbing the wavelength of light that you are seeing. It's saving that wavelength of light for you so you can see it. And we often get confused about this because our interactions with these things in real life make us think like, oh, that thing inherently is green, but what it's absorbing is not green. What it's reflecting is green. Okay. So as you may know, if you've taken physics classes or even chemistry, there are different wavelengths of light, different colors associated with these. Now, plants will reflect green light. So you see that's around 500, and the blending of, green and yellow, really bright green, around 600. So this is what is reflected. So plants are not using these wavelengths of light right here, but they are absorbing these wavelengths of light between 400 and 500, and then oftentimes they're also absorbing from 650 to 700 or something like that. And we know this in particular because we could test plant growth, and we could expose plants to only, or chloroplasts, to only certain wavelengths of light, certain colors of light, and then we get the growth or higher photosynthetic rate at these different wavelengths of light. And then if you give a plant only green light, they don't like it. They will not grow tall at all, and they will look kind of sickly, and they won't be the right color. That is green. The other thing that I wanted to mention is this peak here. So sometimes plants will also reflect some red light, depending on the type of plant, depending on the type of leaf. And that's why sometimes, so I have a lot of succulents, and they sometimes are different colors. Let me just pull one over for a second. So they're not particularly red right now, but these guys both started off pretty red when I got them. And it depends on the type of light that they're getting. So if you put these guys in really bright light, then they will turn red. You can see this one has kind of some reddish brown leaves at the bottom, and this one has reddish brown tinges to the edges of the leaves. And one reason why plants might do that is that they will produce these pigments that will act kind of like sunscreen. And then on the opposite, so that's if the light is too bright. And on the opposite kind of thinking, some plants, if they need more light, will start to produce pigments that can absorb more of that type of light. So shaded plants in particular, they might not be as green, and they might start producing pigments besides chlorophyll so that they could absorb more wavelengths of light because they're not getting as much light overall. And we'll come back to this next week when we talk more about photosynthesis. Just to finish up, this is our diagram for photosynthesis. So again, we start out with light energy. That is absorbed by our chlorophyll. And the light energy is transferred, is absorbed by electrons. And that electron then is going to be excited. And the electron then is going to be passed along between many molecules in the plant cell, many enzymes, and a little bit of the energy from that electron is used to do different reactions. And finally, that is going to turn into bond energy, and in particular, bond energy that forms ATP, or bond energy that ends up in ATP. And what we mean by that is that, of course, what am I doing? Our ATP molecule is going to have some high energy bonds. And so in particular, we've talked about this bond before, and then this bond also a little bit, but not as much as this one here. And if we break this bond, of course, then that is going to create a more stable molecule, and we say that it releases energy. Or what we mean more accurately is that the products are going to be more stable than the reactants, and there's going to be a difference in energy, and that can be used to do work in the cell, to perform other reactions. Okay. However, the problem here is that there's a lot of energy in this photon that is absorbed, and we cannot go straight from light energy to bond energy. We can't even go straight from an excited electron to making ATP. We need to have a lot of steps in between. And it's not just because our cells are inefficient or something, or that we don't have the right molecules to do that. It's because the amount of energy in a photon would be much too high for a single molecule to hold in its bond energy. So essentially, we would get this situation that we see in Toy Story. We would get a ton of energy put in a single bond, and that molecule would be out of control. So I'll leave it there for now. Instead of having this one super high energy molecule, what the process is to kind of distribute the energy over many different molecules. And we'll see how that happens next week when we talk about the reactions in photosynthesis. Okay. So please let me know if you have any questions. I do not have office hours today, but many of you have been texting me at the work number that I set up. You can text me anytime, and I usually will answer those faster than email. So you're welcome to text me there or to email me. And remember, no one has been using this, but I do have my 10 to 8 site where you could set up a private meeting with me where we could talk about class or exam or whatever. If you are missing assignments, which some of you are, please make sure to submit those. And I noticed also that there's a little bit of a bug in Blackboard where some of you started an exam or a test or a quiz or even a homework, and it didn't submit correctly. And so it shows me that you're still working on it, even though it's past the due date. So some of you, I might ask you to try and resubmit those. So just be on the lookout for that email. I don't think it's your fault. I think that it just didn't get through the Blackboard system when you tried to submit it. And so I will send out an email to a couple of you about that. Any questions, comments? Thank you everyone for meeting me here. And I'll also send out an opinion poll on whether you found this to be a more stable system, or Blackboard, or Zoom, which we used last week, in terms of being able to actually see the slides and answer me, things like that. All right, thanks everyone.